This is not a formal note lecture, but this will go over comprehensively different microscope images of epithelial tissues and what you should expect them to look like. And we're going to go over the main common types that were briefly mentioned at the end of the epithelial note lecture, which is stratified squamous, simple cuboidal, simple columnar, simple squamous, pseudostratified columnar, transitional, and stratified squamous. We may also touch on stratified columnar as well as stratified cuboidal, though we will not have as much emphasis on that. So these are the examples that are found in these slides. We'll also look what exocrine and endocrine may look like under a microscope. However, generally speaking, those types of glandular tissues are basically just simple cuboidal or simple columnar. All right, let's go for it. All right, so these are kind of cartoony ways we refer to when we talk about arrangements of layers and cell shapes, but what we really want to look at is what it looks like under a microscope. All right, simple squamous epithelial cells. I'm not a huge fan of this image here, but this is actually a lot more common of an image for simple squamous epithelium, okay, from a above view, generally speaking. Otherwise, if you get simple squamous epithelium, the most likely scenario is you're going to get that fried egg image of separate cells, like that, if they aren't in a clean sheet like this. All right, this is the most common way simple cuboidal looks like, and yeah, I know it doesn't look like squares, so this cartoony image looks more like what you might expect simple cuboidal to look like. But if you look at this image, those don't look like squares. But what actually happens is, because simple cuboidal is most often found in glands and wrapped into circles to form glands, they end up kind of curving into a circle and the squares look more like rounds rather than actual squares. But that's still simple cuboidal, so just a heads up. And this is the most common kind of simple cuboidal when it comes to images in histology. All right, simple columnar, you should see a clear column, okay? So here it looks like columns, and you don't see random cells popping up towards the surface or like a kind of a second layer. Sometimes you see these kind of weird yellow pockets. These pockets are just random cells that form mucus onto the surface. They're called goblet cells, and that's actually fairly normal. So this is a columnar epithelial cell layer, okay? They're not considered ciliated. So ciliated refers to having long hairs. They may have very small hairs, but those hairs are often not fully visible. There would be maybe kind of a light fuzzy layer on the top, but it's not truly all that visible under a microscope. But this is a classic example of simple columnar cells. Cilia, which again is a longer hair that's found on the surfaces of cells, they end up looking like there's like a little fuzzy buzz cut on the cells themselves. But again, you can kind of see it's still kind of one layer. Even though the nuclei aren't perfectly located, it's still like at the same level. It's still pretty clear that this is the columnar cells. The cells underneath of here, notice how they change so rapidly. That's indicating it's a different tissue underneath of it. In this case, connective tissue. So remember, epithelial tissue is often on top of something else. So sometimes there'll be a layer underneath of it that doesn't look like the main layer. But again, you want to kind of just focus on the section that looks like it's part of one type of tissue, one form of tissue, and don't worry about the weird random stuff underneath of it. If it clearly looks like a completely different area, like a completely different section, then you can treat it like that. If it's transitioning, and there is one example of that, or not transitional cells, but if there is one example called stratified squamous where there's distinct weird regions, but it's got a real distinct look to it. And it's still all, those weird regions are still all considered stratified squamous. Okay, so this is pseudostratified columnar. Now, you got a lot of features of columnar, but you'll notice there's random tiny cell looking things here, which makes you almost think it's stratified, but it's not. And the reason why it's not is, generally speaking, when you see stratified, there's really clear layers in stratified. This pseudo-stratified guy has sections where it's one layer and sections where it's two layers, and it's all over the place. That's how you can tell the cells are flopping over and creating weird pockets and weird shapes around each other, okay? So that's why we call this pseudo-stratified. So this is a shape. It looks almost like columnar had a messed up baby with stratified, okay? A simple columnar had a messed up baby with stratified. So it's like, it looks like simple columnar got messed up in a blender. That's what pseudo-stratified looks like. And again, it looks like it's got a little bit of a buzz cut, which is how we know it's ciliated. And generally speaking, when it comes to pseudo-stratified, it's almost always ciliated. Because it's flopping around, it's used for motion, and to increase that motion movement, cilia is going to help extra transport across the surface. Okay. Now let's go into the weirdest guy, stratified squamous. And stratified squamous often gets confused with smooth muscle and with transitional, okay? So, this is an example of what's called non-keratinized, meaning keratinized is found on the skin. This is layers and layers of squamous tissue elsewhere. Now, here's the big thing. Notice these cells all look like they're squashed. They all look like someone was a circle and then they all got sat on. Another thing about stratified epithelial tissue, stratified squamous epithelial tissue, is the layers are thick and there's always kind of weird, darker, denser, rounder layer all the way at the bottom. So if the topmost layers are the most squished looking and it's all irregularly wavy squished, and the bottom's kind of darker, denser, and slightly rounder, that's the sign of stratified squamous. Keratinized is even more crazy because keratinized keeps dead cells attached. So what ends up happening is this whole section is stratified squamous, but this top layer is dead, which is why it looks like kind of almost as though it's a completely separate layer, but in fact this is all epithelial, stratified squamous epithelial tissue. It's just that the topmost layer is super dead and flaky. Okay? But again, you get this, it looks like the top is squished and sat on, and the bottom is incredibly dense and sort of circular, but they're all kind of attached. And that's a sign of stratified squamous. Generally speaking, stratified cuboidal looks like the simple cuboidal, but with more messy layering in that circle. Okay? And stratified columnar, on the other hand, generally speaking, you'll see two layers, and even though it's a little messy, especially on the bottom layer, you can see this top layer is pretty even. And that's how you know it's stratified columnar, okay? And not pseudo-stratified columnar. It's the fact that that topmost layer is fairly even, and even if the bottom's a little messy, that initial layer towards the connective tissue above the basement membrane's messy, you see that nice, clean, rectangular columns. And that's how we know it's stratified columnar. All right. Transitional. A lot of students confuse transitional with stratified squamous because they see many layers. However, the top layer is sort of rounded. It doesn't look squashed. It doesn't look like someone sat on it and squished it. Okay? So, as a result, transitional, this is how I know it's transitional. Because that top layer is sort of rounded and more puffy. It doesn't look like it's squished at the very top, and the bottom doesn't look rounder than the top necessarily. And even when it's stretched, even if these are a little bit stretched out, they're still a little bit curvy. They're still not looking like the absolutely squashed pancake-ness of the top layers of the stratified squamous. So, If you see lots of layers with weird shaped cells and the topmost layers don't look like someone sat on them and squished them flat, that's one way to know it's transitional. So uneven cells, kind of like stratified squamous, but the topmost layer isn't absolute layers and layers of squished cells, transitional. Okay? So that's how we know transitional, this is transitional, versus stratified squamous. And another example of stratified squamous. Even though this is not as squished looking as keratinized, it's still fairly squished at the top compared to the bottom, which you don't see that with transitional. Going over glandular tissues, if you look at this glandular tissue, it's pretty obviously simple cuboidal on the edge. The way I know, though, that it's in particular glandular is because oftentimes with the simple cuboidal stuff, you'll see a lake in the center, and that lake in the center is just collecting secretions. So that's how you know it's glandular, because that empty lake of stuff is the liquid or hormone that's being made and dumped into the center. This is another example of a gland, but again, it's those circles. If you see a circle with a hollow center, and that hollow center is either clear or with a lake of stuff, that's how you know it's a gland. I don't really care if you know the difference between endocrine and exocrine glands. I find that like kind of, we're not at the stage where you really need to know that, but you should be familiar with cuboidal cells are in those typically in that circular shape, and those circular shapes are often forming a gland. Okay? All right, on that note, that ends the review of histology, and hopefully it'll help you better able to identify histological pictures of these various different types of tissues, and if you come across these tissues in organs, you'll be able to identify these tissue types right away. All right, good luck, and move on to the rest of your work for the day. Bye.